Deepali Sharma in Pharmacology Department, M.Pharm Research Scholar. Currently, I am working on pentylene tetrazole-induced epilepsy-like behaviors in zebrafish under the supervision of Dr. ARTI Singh. So today, I will be going to demonstrate about the novel time diving test. This is a standard test which is used to assess the anxiety-like behaviors in zebrafish. Since epilepsy is a neurological disorder characterized by unprovoked and unpredictable seizures, but prevalence of anxiety-like behaviors in epilepsy diagnosed patients are much higher. Therefore, we have used this test to analyze the anxious behavior in zebrafish. Before proceeding to the protocol, I would like to tell you about the apparatus. This is a novel time diving apparatus. This is a trapezoidal glassware with a broader top zone and bottom zone is narrower. We have divided the apparatus into two equal proportions. The upper one is top zone and the lower one is bottom zone. We have filled the water up to 12 centimeter height and 2.5 liter of volume is there. Before proceeding to the protocol, I would like to list up the requirements. So before proceeding towards the protocol, I would like to list up the requirements. And requirements are such as a strainer to transfer the fish from the home tank to the chamber and a normal fish. Another is PTZ treated fish. Here we have administered 170 mg per kg pentylene tetrazole via intraperitoneal injection. Now proceeding to the protocol. Here we will do that. We will transfer the normal fish from its home tank to the chamber, and with same procedure we will also transfer the PTZ treated fish into the chamber of novel tank. Now we will allow the fish to habituate for 2 minutes. Normally, the normal fish will take a dive to the bottom zone and then slowly it will explore the top zone. But in the PTZ treated fish, we can see that the fish will show the anxious behavior as it will remain in bottom zone more than the normal fish. Here we can see that here we have set the timer and we will habituate the fishes for 2 minutes. After 2 minutes, we will record the parameters via a camera that is attached with the laptop, and the video recorded program will be to the 5 minutes. And in that program, we will see the parameters that are such as number 1, the time spent in top zone, the time spent in bottom zone, and number of latencies to reach the top zone by fish, and also we will record that how much entries it takes to the top zone and bottom zone. Here we can see that normal fish explores the whole tank completely, while the PTZ treated fish likes to prefer in bottom zone more. We will be recorded and evaluated these parameters via Animaze software, and these parameters conclude us that the more time spent in top zone will show that epilepsy is that the fish is not anxious, and whereas if the fish spends more time in the bottom zone, it shows that the fish is anxious. And also, if the latency to reach top zone is more in fish, then it evaluates that the fish is anxious. And also, we will calculate the time taken by the fish to reach the bottom zone and the time taken by the fish to reach the top zone. These parameters conclude that if the fish will spend more time in the top zone, then fish is anxious, and Now here we can see that if the fish will spend more time in the top zone, then the fish is not anxious. But whereas when the fish spend more time in the bottom zone, it shows the anxious behavior of the fish. Similarly, if the fish will take longer time to reach the top zone, then the fish shows the anxious behavior. But if the fish takes lesser time to reach the top zone, and it shows that the fish is not anxious, and the phenotype behavior of the fish is intact, which shows that it will cover and explores the whole tank completely. By this test, we can conclude that PTZ treated fish shows anxious behavior, while the normal fish shows very phenotype behavior, that is, it covers the whole tank completely. Thank you.
Good morning everyone, myself Simranjit Kaur, M.Pharm Research Scholar, working under the guidance of Dr. ARTI Singh. Today I welcome you all in the Zebrafish workshop. Currently I have been working on chemotherapy-induced cognitive impairment, which is often called as chemobrain or mental fog. Chemobrain is characterized as loss of speed, forgetfulness, difficulty in learning and memory, and short attention span. So one of the apparatus that we use to the assessment of learning and memory in Zebrafish is T-Maze. In 2001, Downing and Rawling for the very first time used T-Maze for the assessment of learning and memory. T-Maze, as the name suggests, is an apparatus which is built in T-shape. It consists of one long arm that bifurcates into two shorter arms at the angle of 90 degrees. One arm is considered as colored red, and one is colored green. The colored red arm is considered as unfavorable zone, danger zone, or incorrect arm, while the green is considered as safe zone, correct arm, or the favorable zone. So in this apparatus, generally we assess the short-term memory, long-term memory, and memory plasticity with the various parameters. The materials and method required for the analysis of this behavior are T-Maze apparatus, home tank, normal zebrafish, sieve, glass rod, and a stopwatch. So beginning with the protocol, firstly we put the zebrafish in the starting zone and will close the gates. T-Maze, as the name suggests, is an apparatus built in T-shape which consists of one long arm and bifurcate into two shorter arms. One is colored red and one is colored green. The colored red is known as unfavorable zone, danger zone, or incorrect arm, and the colored green is known as favorable zone, safe zone, or correct zone. In the red zone, we give zebrafish the stress, so that will act as an aversive stimuli, and in green we will provide her the food, that is a reward for zebrafish. So beginning with the protocol, for the very first step, we will put a normal zebrafish in the starting zone with the enclosed gates of all the shorter arms and the longer arm also. We will put the zebrafish in the starting zone and will lift the gate and will allow it to explore the long arm for 5 minutes. In the second step, we will lift the gate of the red arm and we will guide the zebrafish to enter the red zone. As it enters the red zone, we will close the gate, and now we will disturb the zebrafish with the glass rod. And the step will be continued for 5 minutes. In the next step, we will allow the zebrafish to enter the green zone. Where the food is present, and that it will be considered as reward. So the gate will be closed for 5 minutes and we will allow the zebrafish to explore it for 5 minutes. While proceeding the experiment, one camera is set up with the laptop which will record the video, and from that video we will analyze the various parameters with which we will be able to know that how much a fish is affected from toxin or how much memory is impaired by giving the toxin. After recording the video, we will analyze it with the software enemies, from which we will analyze the parameters such as transfer latency or escape latency, the time taken by the zebrafish from starting zone to reach the green zone or the safe zone, and number of entries in the green zone, and the total time spent in the green zone. From all these parameters collectively, we will able to know that how much memory is impaired by administering a toxin, and we can differentiate it with from a normal fish behavior. And lastly, I would like to conclude that TMA is not only for memory learning and assessment. It is also used earlier for the color preference in zebrafish, that which color a zebrafish prefers more. Along with that, in some experiments, or it is reported in literature that it has been used for the assessment of anxiety-like behavior also. So this was all about TMAs, and thank you very much.
Good morning to all. Myself, Lakshay Kapil, MPharm Research Scholar in Department of Pharmacology from ISF College of Pharmacy, MOGA. Currently, I am working on chemotherapy-induced cognitive impairment under the supervision of Dr. ARTI Singh. I welcome you all in a IBRO-sponsored zebrafish workshop. Now, today, I am here to demonstrate our design patent, which is designed by our team, that is stress maze. As stress maze, as you can see, Y in shape, where long arm is divided into 4 different compartments, each containing a different type of mechanical stress. After that, it subdivided into 2 parts, that is, one is darker zone and another is lighter zone. Inducing the stress in the zebrafish, we have to explore the zebrafish into the 4 chamber. Firstly, we have to put the zebrafish into the first chamber, that is a high speed aeration chamber. We put the zebrafish into the high speed aeration chamber for 3 minutes. After 3 minutes, we have to open the slit and transfer the zebrafish into the second chamber, that is a pool hurdle chamber, in which the movement speed is disturbed, or moving pattern is disturbed of the zebrafish. After 3 minutes, we have transferred the zebrafish into the third chamber, that is a predator chamber, in which the zebrafish feel stress by seeing the another type of animal. After the 3 minutes, we again transfer the zebrafish into the fourth chamber, that is a glass rod chamber, in which we have give the stress by using a glass rod. In glass rod chamber, we have transferred the zebrafish into the resting chamber for 1 minute, and here the zebrafish rest for 1 minute. After that, we have to open both the slits of this chamber, and we have seen that where the zebrafish is gone. If the zebrafish first gone to the darker zone, then it will show that there is a depressive behavior of zebrafish, and if the zebrafish goes to the lighter zone, then it will show there is a normal behavior of zebrafish. So now there is a question, how to differentiate between stressed fish and a normal zebrafish. So for the normal fish, we have to first explore, we have to put the normal fish into the resting chamber for 1 minute. After that, we have to open the slit of light zone for 2 minutes and explore the lighter zone for 2 minutes. After that, we again transfer the zebrafish into the resting zone and open the slit of darker zone, in which we have to explore the zebrafish for 2 minutes. Now, so now for the stressed zebrafish, we have to explore through all the 4 chambers, and after that we have to put it in a resting area for 1 minute, and then we open both the slit of both dark zone and lighter zone and allow to explore the zebrafish in both the zone for 2 minutes. The total duration of this test is 15 minutes, as 3 minutes in light, 3 minutes in high speed aeration, 3 minutes in pool, 3 minutes in predator, 3 minutes in glass rod, and 1 minute in resting area. After that, the 2 minutes of exploration in a darker zone and the lighter zone. The video recorded by Anime software, and we have to analyze this by exploring the chamber in which zone, as they prefer the darker zone or the lighter zone. If the zebrafish prefer the darker zone, then it shows a depressive nature, and if it prefer the lighter zone, then it shows a normal behavior. So this is all about the stress maze. Thank you.
Good morning everyone, this is Bhavna pursuing M Pharmacy under the supervision of Dr. ARTI Singh in Neuroscience Division, Department of Pharmacology from ISF College of Pharmacy, MOGA, Punjab. I welcome you all in this Evro-supported Zebrafish workshop. Today I am here to demonstrate about open field test. Now proceeding about the introduction about open field test. The open field apparatus was firstly developed by Kelvin S. Hall in 1934. An open field apparatus utilizes a large cube box and the top portion of the apparatus is typically left uncovered. In scientific research, the main purpose of this test is to assess the exploratory behavior, general locomotor activity level and anxiety-like behavior in rodents as well as zebrafish. Parkinson disease is a neurodegenerative disease characterized by a loss of dopaminergic neuron in the compact part of the substantia nigra. It is characterized by various symptoms such as bradykinesia, stiffness, muscle rigidity and tremors. Since Parkinson disease is associated with locomotor activity, so I have used this apparatus in my study to assess the locomotor activity. Now I want to list the requirements to perform the open field test. We have need a zebrafish and pre-filled open field apparatus filled with water up to 70% and sieve to transfer the zebrafish from home tank to open field apparatus, and we have also need a stopwatch to measure the time. Let's start with the protocol. Firstly, we will take a zebrafish from home tank and transfer into open field apparatus. Let it to be habituated for 2 minutes. After habituation, we will set up a camera for recording, and recording will be done for 2 minutes. After recording the video, we will analyze the video by Animage software and we will observe the parameters such as total distance travelled, average speed to analyze the locomotor activity. And it is well reported in the previous literature that the normal fish explore the more open area of the apparatus, but the fish treated with toxin doesn't explore more open area of the apparatus. So we can conclude that the open field apparatus is very useful for the assessment of locomotor activity. So this is all about the open field apparatus. Thank you.
Now I'll be demonstrating about the light and dark chamber. Light and dark chamber is basically used for the assessment of anxiety-like behavior in zebrafish. So it is a rectangular shaped instrument which is made up of plexiglass, and here it is divided into 2 equal compartments. One is covered black and one is covered light. Okay, so for the partition we use a slit, and which is also black in color. This is for the partition, and we fill up to 80% of the tank with water. The water should be similar as of home tank, including all the medicines that are included in home tank. And after that, after filling the water, after filling the tank with the water, we will transfer the zebrafish into the dark zone, and the slit will be closed, and we will start the recording and will allow the zebrafish to acclimatize in the dark chamber for 1 minute. And here you can see that the zebrafish is moving and exploring the tank, and the procedure will be continued till 1 minute. And while doing this, we will attach a camera with the laptop for the recording purpose, which we will analyze with Animage software, and from that software we will include count our parameters for the assessment of behavioral activity in normal zebrafish, or same procedure is followed with any toxin or chemical treated zebrafish. So why we give it a 1 minute of habituation? Because zebrafish habituate itself very easily in any new environment. So 1 minute is enough for its habituation. As you can see that only few time is left. So the time is completed, and now we will remove the slit and will allow the zebrafish to move freely or explore the whole tank. And in the normal fish tends to spend more time in light zone because it has no deformities or no any stress or anxiety like behavior. So it remains in light area only, as here it is seen in that even after the habituation of 1 minute in darker zone, it still prefer to move into the light zone, which shows that this fish is normal. And same parameter or same behavior analysis we use for the assessment of anti-anxiety drugs, where after giving stress or anxiety to zebrafish, we check whether the drug is effective or not. For the analysis of parameters, the parameters which we assess are, with the help of anime software, while after recording the video, are number of entries in light zone, number of entries in dark zone, total time spent in light zone and total time spent in dark zone, and latency to enter the light zone first are recorded with the help of anime software. So in conclusion, I would like to say that this apparatus is very well established and very useful for the assessment of anti-anxiety drugs, in which after giving the stress or anxiety to zebrafish and then by providing them a treatment, we can assess whether the fish is now anxiety free or not, and that could be a good approach for the behavior analysis. Thank you.
So now this is our newly designed patent that is designed by our team, which is a learning maze. In this, this maze is basically made in square in shape and have 10 centimeter height and the level of water up to 70%. So as you can see that this is our learning maze apparatus, in which it is in zigzag form. The zebrafish follow our zigzag pathway to reach their platform. As you can see that the zebrafish follow a zigzag pathway and they move in a zigzag pattern. Help of glass rod, we move the zebrafish, and so as you can see that firstly we have to put the zebrafish into the reward section for the habituation for 1 minute. In the reward section, we give the feed to the zebrafish. So now in the test session, we put the zebrafish in the first chamber and we open the slit of the target zone, and we see that the zebrafish follow the pathway in the zigzag pattern. As you can see that the fish goes to their target zone, and in this we have total check the latency time, how much time the fish take to reach their target zone, and we in this apparatus is basically used for the cut. So now we conclude that in this apparatus we've checked the total time for the zebrafish to reach, taken by the zebrafish to reach to their target zone, and this apparatus is we used for their learning maze, and in this apparatus is used for several and this apparatus we used for learning maze, learning. So this apparatus is used for assessing the learning memory in several neurological disorders like Huntington disease, Parkinson disease, Alzheimer, epilepsy and chemobrain. Thank you.
So now I'll be demonstrating about the plus maze. It was a design patent made by our senior Mr. Sukhdev Singh. As you can see, it is composed of a forearm in plus shape. It contains 4 chambers. One is enclosed with mirrors inside it, and while other 3 are transparent. The height of the chamber is 12 centimeter, and with 10 centimeter we fill it up with the water, and then we proceed for the protocol. Before proceeding towards the protocol, as you can see, this apparatus is a fusion of 2 models. One is mirror chamber and one is T-maze. That is why it can be used in multiple models of behavior analysis in zebrafish. So for the assessment of anxiety-like behavior in zebrafish, we will put the zebrafish in this mirror chamber, as it is containing mirror in this interior walls, and fish will get anxious by seeing her own picture, and there will be induction of anxiety or aggressive nature in the zebrafish. We will record this behavior with the help of. After placing the fish in the mirror chamber, we will allow the fish to stay in here for 5 minutes, and same time recording will be done, and from the recording we will conclude speed of fish and number of biting, and then we will transfer the same fish in the novel diving tank apparatus also and in light dark chamber to check whether the anxiety has been created in zebrafish or not. Also, as I already explained, the other arms of the plus maze are transparent, with which we can covered with different colors to check the color preference of zebrafish, as zebrafish attracts to color very positively. So with that we can check the color preference, and also this can also act as a T-maze where we can assess the memory and spatial learning of zebrafish, and this long arm is we can use this for the locomotion activity of zebrafish in this long arm. So after the complete demonstration of the apparatus, from this we can conclude that this apparatus can be used for various neurological disorders, such as for locomotion, where in Parkinson disease or Huntington disease where locomotion get affected, we can assess the locomotion. Chemobrain, epilepsy, Alzheimer's, where memory gets affected, we can check the memory. And for anxiety-like behavior, we have here is mirror maze. So from that, this maze is a multiple usable apparatus with which number of behavioral analysis could be done. Thank you.
Now I will be demonstrating the sex difference between male zebrafish and female zebrafish. We have separated the fishes with respective tank. This is for male and this is for female. And for better visualization, we have separated out the zebrafishes into different beakers respective to their tanks. So on the basis of the color, male zebrafish are more yellow and pink in color, whereas female zebrafish are more whitish and bluish in color. And if we talk about the attractiveness, male zebrafish are more attractive with a colorful stripes, but female are less attractive. On the basis of their body shape, males are cylindrical in body and females have bulgy portion at their belly portion, because the female zebrafish have to accommodate the reproductive organs that are ovaries and others. For better visualization, I would like to focus on the male zebrafish. The actual difference between the sex is known about the age of 3 months. And these fishes are also above than 3 months. So here we can clearly see that if we talk about the body, then this is cylindrical in shape, and I talked about the color. Here we can see that they are more yellow. Similarly, on the basis of color, we can clearly see that this is a female zebrafish, and in this the color is more bluish and whitish, and the belly portion is more bulged out. Here you can see the clear difference between male zebrafish and female zebrafish. Here is the brief introduction about zebrafish life cycle. So here we can see that this is the first stage of life cycle, that is fertilization. 0 hours post fertilization. After 50 hours of post fertilization, the fertilization will occur into embryo. Then after 15 days post fertilization, embryo is changed into larva. Then metamorphosis occur after 50 days post fertilization. And in metamorphosis, organogenesis occur, in which liver, kidney, heart, these organs develop. Then after 28 days post fertilization, the stage comes, that is juvenile stage. It is the infant stage of zebrafish. Then after 45 days post fertilization, the zebrafish converts into puberty. That is the age in which it is capable of spawning. Then after 60 days post fertilization, the zebrafish becomes adult, and again So here is a brief introduction about zebrafish life cycle. We can see that this is the first stage, that is fertilization stage. Here after 0 hours post fertilization, it changes into the embryo. Then embryo took 50 hours post fertilization, that is equal to 2 to 3 days, to convert into larva. Then the next stage comes, that is metamorphosis. It took approx 15 days post fertilization to organogenesis, in which organs like kidney, liver, heart generate. Then after 28 days post fertilization, juvenile stage reaches, and then puberty stage reaches after 45 days post fertilization. Here the spermatogenesis and gametogenesis occur, and then after 60 days post fertilization, the zebrafish is reached to the adult stage. Here the zebrafish is capable of spawning, and it is able to produce gametes, and the spawning goes on throughout the year. And zebrafish is also capable of laying 500 to 700 eggs per spawning, and the spawning can go out throughout the year, and here the zebrafish life cycle goes on.
For fish culture and establishment of fish lab, the water should be fresh and should be in abundant amount, and it should be free from any kind of contaminant and pathogen. It could be any fungal, any microbe, or any virus. To avoid that things and to make the fish free from any kind of disease, we use certain medicines which are anti-itch, then anti-chlor, and then antifungal. We use these three medicines to make the water good for the zebrafish facility. To maintain the better condition, the pH and temperature should be checked thrice a week, and washing could also be done thrice a week to maintain the good and healthy conditions for living of zebrafish. Apart from this, feeding is an important aspect for the fish culture and zebrafish establishment. So here we use tetrabite fish biscuit for the regular feed. As you can see, the size of feed is larger, so we triturate the feed into very small particles, and then we put in the fish tank, so that the fish should not engulf the larger particles and they do not die. So these are our food items, and for the weight gain we use extra feed biscuits. These are dried worms for the weight gaining of the zebrafish, to make them healthy, so that we can perform our experiments on them according to the CPCSA guidelines. So now I will discuss what are the basic similarities, what are the anatomical similarities with human beings and zebrafish. So now here we can see that in this poster that the zebrafish has heart, spleen, kidney, both head and body, then muscles and testes, and in female there are ovaries. So these are the basic anatomical similarities with humans and zebrafish. And why? And these are the reasons that zebrafish is becoming a popular and favorable model of biomedical research. And not only in this, we can also show us the similarities between the human brain and zebrafish brain. As you can see there are organs which are present in human brain are also well defined and well identified in zebrafish brain, such as olfactory bulb, telencephalon, then hippocampus for cognitive sciences. And not only in adult zebrafish, these organs are also well established in larvae. Here the telencephalon, then brain stem, then tectum opticum, which will later on develop into a fully functional brain.
Going to demonstrate about the breeding chambers. We have two different breeding chambers. Firstly, I would like to tell you about this chamber. This is a breeding chamber we conventionally used, in which there is a open lid, this is a slit, and this is the base portion, which is divided into two parts. Here the eggs are collected, and at the above part the zebrafish will remain there only. Here we put the water that is maintained in the home tanks only. Then we will put the chamber for incubation. We can place this inside the tank by putting the double tape and like this, and we will maintain the 14 and 10 hours dark and light cycle. And after putting the male and female zebrafish, that is ratio of 2 females and 1 male for increasing the probability of spawning, we will provide the proper environment, and at the end of light dark cycle, that is after 13 hours, we will remove the slit and allow for mating. Then after 1 hour, we could collect the eggs from the bottom portion. This is the separating portion, because what is observed that zebrafish usually eat their eggs by self only. So for preventing the self eating of eggs, we collect the eggs into bottom portion. Then we will collect the eggs via siphon tube and wash them with methylene blue water, and this water is used for preventing the eggs from various infections and other contaminations. Here we have the another breeding chamber. This is also like a previous one, but in this chamber we had the facility of hanging it outside like this, and as well as inside the home tank like this. Okay. The other facility that we could have in this breeding chamber is that we have aeration facility, and there is a attached bottom portion which we can remove easily. We found this chamber more convenient and easy to handle, because in this we could place the chamber inside or outside the home tank, and here we have the facility of aeration also. Major drawback of previous chamber is that it get into the home tank and the whole setup get destroyed. Other major disadvantage that we found is that in this zebrafish goes down and eat its egg self, but in this there is no space for zebrafish to enter into the base. Both the chambers are available at Amazon. You could find it very easily. Thank you.
Good morning everyone, this is Vishal Kumar. Currently I am working as a junior research fellow in DST-SERB sponsored project under the supervision of Dr. ARTI SINGH in Neuroscience Division, Department of Pharmacology, ISF College of Pharmacy, MOGA, Punjab. I welcome you all in this IBRO supported meeting and Zebrafish workshop. So today I am here to demonstrate to how to weigh the fish. So we need to some accessories. First of all, we need to a normal tank which contain a normal fish, and for the anesthesia either you can use ice water or 0.2% trichine solution. Later on we need a empty petri dish, and you need to a weighing balance. So how we start? First of all, you have to switch on the apparatus and then put a empty petri dish on a weighing balance and then tear it, which shows 0. Here is the unit is in mg. Okay, so let's weigh. This is the anesthetized zebrafish. I am transferring in a empty petri dish which is already teared, and now I am put it on the apparatus. So the weight of this fish is showing 310 mg. It means this fish has a 310 mg weight. Thank you. Before proceeding the protocol, you have to prepare yourself as per the CPSA guidelines for the zebrafish experimentation. So how you can prepare? You have to wear a proper gloves, you have to wear a mask, you have to wear a head cap, and you also have to wear a apron. You have to need some accessories or materials. The first one is a normal tank which has a normal fish. Second one, you need a sieve to transfer the fish from the normal tank to the anesthetic agents. This is the anesthetic agent that we are going to use. This is the sharp forceps. Sometimes you have to need the fish from the anesthetic tank to the sponge. This is the sponge. You can see that here is a cut that is 3 centimeter long. It will depend upon the length of the zebrafish. It may be 4 centimeter, it may be 5 centimeter, okay. And this is the normal saline, and later on this is the Hamilton syringe. You can see that the maximum volume in this syringe you can fill 10 microliter, which have a sharp needle. For the convenience, this is pre-filled with a normal saline. So let's start the protocol. First of all, I will transfer the normal fish from the normal tank to the anesthetic tank, which is already have a anesthetic agent. So you can see that now the fish is anesthetized. But the thing is that how you identify the fish is anesthetized? You can identify that if the movement of fish is completely slowed down, but the gills of the fish is still in working, it indicates the fish has been anesthetized. And immediately take a fish and transfer it to the sponge. Now immediately within a second you have to inject the fish. The point is that what is the where is the IP cavities shows. You have to inject just anterior to the anal fin of the zebrafish. You can see that this is the anal fin because this is the belly part. This is the anal fin which is used for the balance of the zebrafish. Now I am injecting it just anterior to the anal fin, and then now it is completely injected. After the injection, immediately transfer it to the normal tank. It will take 8 to 10 seconds. After the injection, immediately as I have transferred it to the normal tank, it will take 8 to 10 seconds to normalize. I hope this video or this demonstration, this is all about IP administration of This is all about the intraperitoneal administration of drug, chemicals, toxin into the zebrafish. Thank you. Now I am demonstrate the intramuscular injection in a zebrafish. How you can inject a drug or toxin in the zebrafish with the help of or via the route of intramuscular. So as for the convenience, I have already anesthetized this fish and immediately I have transferred it onto the sponge. Immediately I have put the fish on the sponge. Remember one thing, if you are going to inject intramuscularly, you have to expose the dorsal part of the fish. But if you are going to inject intraperitoneally, you have to expose the belly part. So now we are going to inject intramuscular injection. So remember one thing, this is the dorsal fin. You can see that this is the dorsal fin which is used for the balance their body. Just you have to inject just anterior to the dorsal fin. This is the whitest portion. So in this portion you have to inject. This is I am injecting, and then press the plunger of the Hamilton syringe and immediately transfer it to the normal tank. Till now I have demonstrated two different routes of drug administration. First one is a intraperitoneal injection, and the second one is intramuscular injection. Apart from this, as per the CPCSA guideline, you can also use a different different routes of drug administration. For example, cerebroventricular microinjection and oral administration. Thank you. As previously we have discussed so many things about the zebrafish, and now I am going to demonstrate, I am going to dissect the different organs from the male as well as female zebrafish. Before proceeding the dissection, let me introduce what are the materials we have required. This is the male zebrafish which is used to dissect the testis, kidneys and swim bladder. This is the female zebrafish which will use to dissect the ovaries, heart and brain as well as intestine. This is the PBS solution and this is the ice cold water which is used to anesthetize the fish, and these are the dissecting pins which will help to stable the fish on the dissecting mat at the time of dissection. This is the PSS solution which will be used to see the heartbeat of a zebrafish, and this is a venous spring scissor which is used to cut the zebrafish, and these are the sharp forceps which will use to remove the extra tissues, to expose the belly parts, and this is the empty patty dish which will be used to remove the extra tissues from the particular organ. This is the simple microscope which will be used to visualize the organ, and this is the sieve which will be used to transfer the fish into the ice cold water to anesthetize that fish. And this is the dissecting mat which will use to stable the fish. Let's start the protocol. First of all, we will start from the male zebrafish followed by female zebrafish. So firstly, you just remove the male zebrafish from the normal tank or from the home tank and then transfer it to the ice cold water to anesthetize that fish and wait for the 4 to 8 seconds. That in this time this fish will completely anesthetized. Once the movement of fish is completely stopped, then we can remove the fish from the ice cold water. Start. After the anesthesia, you have just removed from the ice cold water and then put it on a dissecting mat, and the first process is that to tap dry the completely fish. Once it dry, then you have to put a dissecting pins, one pin at the back side between the caudal fins. Now I have fixed it, and the second pin you have just put just anterior to the eye cavity. Now the second fin is also fixed, and let's start the dissection. You can completely see that the first pin is just put anterior to the eye cavity and the second pin just between the two caudal fins. Now I am going to make a cut. The first cut is to make just anterior to the dorsal cut, let me introduce the outer anatomy of the zebrafish. You can see that these are the two caudal fins and this is the dorsal fin. Here is a dorsal fin. So here is a dorsal fin which will use by the fish to balance. At the belly side, this is another fin. Here is two fins. One is you can see that at the belly side it has a pectoral fin that is near to the gills and a pelvic fin just back to the belly, and one is a anal fin just anterior to the anal cavity. Okay, let me cut. So the first cut I will make just anterior to the anal fin. You can see that, and gently cut the total belly part up to the pectoral fin. You can see that. And the second cut just make to expose the completely gills and remove that extra part. You can clearly see that I am making that completely cut, and now you just expose the complete belly part. Here you can clearly see that some internal organs, these are the some internal organs. Okay, now I'm going to remove the testis. You can see that this is the white color portion is a testis. You can see that now I have removed the complete internal organ from the zebrafish and then transfer it to the PBS solution, and you can see that this is the whitish color portion is a testis. Now I am going to visualize this portion in a simple microscope. You can see that this is a fused organ, that these are small particles. Now I am going to dissect the brain from the zebrafish. For that you have just remove the pin and turn the fish at the side that the belly part is at the upper side and the dorsal part is in a bottom side, and just put a pin and then completely remove. After removing the pin, you have just cut a complete head part and separate out from the body part. So here is the body part is separated, and then just it put on another dissecting table or another dissecting mat. With the help of sharp forceps, you have just remove the extra tissues from the head part to expose the skull. Once the skull is exposed, now I am removing the extra tissues from the head part. So there are so many tissues as present, so it could take a time to remove the extra tissues. Now I am feel a hard portion, that is a skull. We all know that the brain of zebrafish is entrapped in the skull. So firstly we have to expose the skull to see. Now the tissues has been removed. Now the extra tissues has been removed and skull has been exposed. You can see that this is a white portion. This is a brain. And now I am going to remove some more tissues. I have just take this head part and place in a PBS solution. It will help me to remove the complete brain. So I am gently remove the extra tissues and the skull part. It can break the entire brain. Now I have completely removed the brain part. You can see that this is showing some reddish color. To visualize the more, okay, for the clear vision I have put it on a simple microscope. You can see that this is the upper portion. It is a telencephalon, and these are the some belly portion at the both side. This is a tectum opticum, and after that it has a hypothalamus and then cerebellum. Now moving on, the female zebrafish, we have just put it in a ice cold water to anesthetize as described previously, and left it for 4 to 8 seconds. After 4 to 8 seconds, the fish has completely anesthetized, and just put it on a dissecting mat. Expose the belly part as described previously. Just cut from the anterior part to the anal cavity or anterior to the anal fin. Start. After exposing the belly part, I have just removed the internal organs and put it in the PBS dish. If you focus this portion, this is a heart of the female zebrafish. If you focus, you will see the heart beating. This is the atrium and this is the ventricle part. You can see the proper beating of the heart. After the dissection of heart, let's move on the dissection of liver. You can see that This is the liver which is reddish color, which is appeared in a reddish color and attached or accumulated just after the dissection of liver. Let's move on the dissection of GIT system. You can clearly see that this is a small intestine. To clearly see the length and the particles that are entrapped inside the intestine, I have just put it on a simple microscope. Now, to clearly see the length of the small intestine and how it is visual on a large scale, you can see that this is a reddish portion that you can see this. This is a gallbladder, okay, and rest of it, this is a small intestine. This is clearly seen. After the dissection of gallbladder and small intestine, let's move on the dissection of ovaries. So for the clear visual, I have put a ovary at the simple microscope. You can clearly see that these are the small particles. After the dissection of small intestine and gallbladder, we have moved to the dissection of ovaries. For the clear visual, I have just put it on a dissecting microscope. You can see that this is a small round shaped particles are the eggs. For the clear visual of swim bladder, I have put it on a dissecting microscope, and you can see that this is a fused organ. One is a small and second one is a larger than the first one. So this is all about the dissection of zebrafish from female as well as male zebrafish. Some of organs we have removed or we have dissected from the male zebrafish, and some organs are dissected from the female zebrafish. Thank you.
So finally, in this workshop, we have demonstrated about the neurobehavioral aspects in zebrafish, how you can use zebrafish for various behavioral aspects. Apart from this, in this workshop, we have also demonstrated about the different routes of drug administration, whether it is IP, whether it is oral, intramuscular, intracerebroventricular, whatever route it is, we can administer the drug, whether it is toxin or any drug, we can administer. Also, we have demonstrated about the different organs, how can you isolate different organs from zebrafish. Apart from this, we have also shown you the beating of heart in zebrafish. I hope this workshop, it would be helpful in the future of all those researchers who want to work on zebrafish. I would like to thank International Brain Research Organization for providing us the support. I would also like to thank Chairman ISF College of Pharmacy, Shri Parveen Garg ji, Director ISF College of Pharmacy, Dr. G.D. Gupta, Vice Principal, Dr. R.K. Narang, for providing us the research facility for performing these research work at this institute. I would like to give special thank to Dr. Charan Singh, Head to the Department of Quality Assurance, who have given us this opportunity to show our work at this international platform. We really thank me and my team members, including PhD research scholar Mr. Vishal, MPharm research scholars Mr. Lakshay Kapil, Ms. Simranjit Kaur, Ms. Deepali Sharma and Ms.Bhawna. We would like to give special thanks to Dr. Charan Singh, Head to the Department Quality Assurance, for giving us this opportunity to present our research work at this international level. Apart from this, we would also like to thank all the participants, all the delegates who are watching this workshop. Thank you very much.